My name is Jessica Durston. I'm going to be speaking to you today about high throughput screening for drug discovery, specifically what it is and how it is being used, what are the advantages and what are the limitations of using this technique. Traditionally, drug development is expensive and can take a very long time. High throughput screening was developed in order to speed the process up and to save money. High-throughput screening is the use of automated equipment to rapidly test thousands to millions of samples for biological activity at the model organism, cellular, pathway, or molecular level. High-throughput screening was first developed in the 1990s. Since then, there have been huge technological advances in assay miniaturization, automation, and robotics. A related technology to high throughput screening is called high content screening, or HCS. In contrast to traditional HTS, which has a single readout of activity, HCS allows a scientist to measure many properties or features of individual cells or organisms at once. High content screening, or automated microscope-based screening, measures biological activity in single cells or whole organisms following treatment with thousands of agents such as compounds or siRNAs. HTS and HCS employ different technologies, including sample preparation, automated analysis equipment, supporting detection reagents, data processing software, and bioinformatics. HTS falls under early drug discovery in the drug development pathway. This is just an example of an HTS workflow. It is an overview of antimicrobial screening. Bacteria were inoculated with a colony picking robot, either in monoculture or one-to-one interactions on plates. For the detection of antimicrobial activity, an agar overlay assay with 2 target organisms was performed. Antimicrobial activity was determined after overnight incubation by screening for visible zones of inhibition in the upper agar layer. HTS can involve many different assays and methods discussed in MIP 611, including immunohistochemistry-based assays, cell-based fluorescence reporter gene assays, cell-based assays for detecting protein-protein interactions, protein fragment complementation assays, or PCAs. As shown in the diagram on the right, in addition to cell-based assays, there are also in vivo HTS assays, as well as biochemical assays. HTS assays use 96 well, 384 well, or larger plates. Control wells are built into the plate map and into the experiments. Assays are often repeated to ensure reproducibility. The middle picture is an example of an HTS drug screening plate setup. The assay design is optimized for a 384 well plate and for 10-point drug dilutions. The most efficient design is to use 2 blocks of 16 drugs, leaving 4 columns available for negative and positive controls. This allows 32 drugs to be tested on a 384 well plate. The figure on the right is showing how a plate was set up for screening anti-HCoV OC43 compounds. This paper was looking at high throughput screening and identification of potent broad spectrum inhibitors of coronaviruses. When compared to traditional or non-high throughput screening techniques, HTS has quite a few advantages. It has high sensitivity, including single molecule detection. It is high speed because it's automated. It has a minimization of samples because of microtiter plate assays. There is simplicity, there's rapidness, there is a lower cost from using fewer reagents, among other reasons, and high efficiency. The figure on the right is showing the dose response of the initially identified 4,166 selective hit compounds. This is just an example of how many hits can be generated using HTS. In this particular paper, over 1.8 million compounds were screened. Some limitations to this technique, which will probably be more obvious as time goes on, are the possibility of false positives, data storage limitations, and data transfer between platforms. Thank you for taking the time to listen to me.